processes. Now, complement system. Complement system is a system of soluble components of innate immunity. It consists of C1 to C9, 9 protein enzymes which enhance inflammation. Enzymes of the complement system are serine. So as a result of antigen-antibody combination, there is cell lysis, and this is enhanced by complement system. So what happens in complement? WBCs are attracted to the site of the complement system, and pathogens are phagocytosed by neutrophils and monocytes and macrophages. Complement activation is a critical component of innate immunity. 4 pathways of complement. Number 1, classic activation pathway, which is dependent on antigen-antibody reaction and is activated by C1, 4 and C2. Alternative pathway, antibody independent, activated by microbes or tumor cell by binding of C3 directly to the pathogen. For example, in immunoglobulin A nephropathy, IgA activates alternate complement pathway, causing glomerular damage. Number 3 is the activation pathway, activated by microbe. And number 4, terminal pathway, a combination of the above 3 that leads to cell lysis. So the complement system consists of 9 enzymes from C1 to C9, and the key factor is C1, which has 3 subunits, C1q, C1r, C1s, in both innate and adaptive immunity. So what's the function of C1? C1 recognizes bacterial and viral sugar structures which are different from a cell. Interaction of C1 with antigen activation produces holes in the antigen that leads to leaks in the cell, causing cell lysis. C1 binds to the immunoglobulin that is bound to antigen. So it binds to the immunoglobulin that is bound to antigen, and it triggers a reaction that activates C3 and C5 and converts them into C3a and C5a, which are the active form, and that releases histamine from granulocytes, mast cells and platelet, leading to vasodilatation and increased capillary permeability. So the basic function is to increase capillary permeability by vasodilatation, and this is done when C1 binds to antibodies, immunoglobulins, which are already bound to antigen. That causes activation of C3 and C5 and converts them into C3a and C5a, and that releases histamine from granulocytes, mast cells and platelet, leading to vasodilatation and increased capillary permeability. C3b causes opsonization, and C5a, C5b, C6 and C7 are chemotaxis and attract WBC at the site of antigen-antibinding reaction. C3 cleavage by first 3 pathways activate C5, C6, C7, C8 and C9 that leads to cell lysis. So complement is a system of soluble protein of innate immunity that has serine protease. When antigen combines with antibody, to this antibody the complement attaches, and that attracts the WBCs, and pathogens are phagocytosed by neutrophil, monophage and macrophages. There are 4 paths. Number 1, classical activation pathway, antigen-antibody dependent. Alternative pathway, antigen independent. Activation pathway, activated by microbes. And the 4th pathway is a combination of the 3 that leads to the cell lysis. The first step or the key factor in the complement is C1, which recognizes the bacterial and viral sugar structures, and C1 combines with the antibody that is attached to the antigen and makes holes in the antigen by causing chemotaxis and attracting neutrophils, monophage and macrocyte at the cell. C1, when combined with immunoglobulin, activates C3 and C5, and that releases histamines from granulocytes, mast cells and platelet and causes vasodilatation and increased capillary permeability. Now these enzymes, as I already told, have protease. So what substance inhibits protease activity? C1 esterase inhibitors inactivates protease activity of C1 in the classical pathway, and C1 esterase inhibitor deficiency causes hereditary angioedema. What disorders occur in different types of deficiencies of the components of complement? In the deficiency of C142 classical pathway, infection with pyogenic bacteria occurs. Number 2, in the deficiency of factor B, And the alternative pathway, increased neisserial infection occur. Number 3, the deficiency of C3, there are recurrent bacterial infection. And in the deficiency of C5, 6 and 7, there occur recurrent meningococcal and gonococcal infections. And number 5, paroxysmal nocturnal hemoglobinuria. We'll discuss it in a separate video. Now how the complement is tested. Decrease in C2 and C4 indicates activation of the classical path, and decrease in factor B indicates activation of alternative path, and decrease in C3 indicates activation of either. So decrease C2 and 4 indicate classical pathway activation, decrease factor B indicates alternative pathway activation, and decrease C3 indicates activation of either.